Let's continue our romp through the microarray literature, now looking at examples of how microarrays can help affect improved prediction of clinical outcomes. The paper here that we're discussing is Van Veer from 2002, Nature, and they're looking at the prognosis of breast cancer metastasis. This is relevant since the metastasis, the status of the tumor, can result in the prescription of adjuvant therapies. And these therapies, of course, are designed to slow or prevent the progression of the cancer to metastasis, but they also come with significant side effects, which we won't go into today, but so suffice it today to say that on the one hand, we want to avoid the side effects if possible, and two, we want to avoid incurring the costs of prescribing those adjuvant therapies if necessary. It's also worth pointing out, as the authors do, that back in 2002, it's getting on in years now, some 90% of non-metastatic breast cancer patients were considered good candidates for adjuvant therapy. So almost as a preventative measure, they're assigned adjuvant therapy. All right, now that's an important point that we'll come back to again at the end. First of all, let's look at the data here. We have an oligonucleotide array probing some 25,000 human genes synthesized in situ. So there's our connection to the methods portion of this. As opposed to being inkjet printed on the slide, these are synthesized one nucleotide at a time on the in situ on the slide. Assembling some 117 different breast cancer patients, and they're ranked according to expression pattern. And here, the ranking is whether or not they are, I think these, above the yellow line is ER positive, that is to say estrogen receptor positive, and below the line is BCRA1 positive patients. That's according to, that's according to histochemistry. In any case, tumors are classified according to their prognosis, short or long interval metastasis, that is to say less or greater than 5 years. And then so we try to make sense of the expression profiles that we see. And there were some 231 genes that reported on the metastatic state of the tumor, and of these they chose for a panel as a predictor 70 genes, and they were able to assign tumors to the proper category with a very high success rate. So this is pretty powerful because it allows us to say, based on the gene expression pattern of the tumor, whether the tumor is likely to go metastatic in the near or long term. Okay, again, we're not looking at the genes that are, in this case, we're not going to look at the genes that are relevant here. I'm not going to talk about that. Instead, I'm going to talk about the fact that what we're really looking for is a way to classify this tumor as something that's a good candidate for adjuvant therapy or not. And according to their analysis, over 70% of the patients in their study were probably going to be prescribed the adjuvant therapy unnecessarily. So I don't know that this method has been used to change the treatment practices for breast cancer. It would be interesting to find that out. Hint, hint, discussion questions, etc. But I think it's worth pointing out that the tools are in place for using genomics to change cancer therapies. Let's turn now to protein arrays, and let's see that we can use protein arrays to understand or to probe and understand adverse drug reactions. In this case, we're looking at antifungals. There's a few treatments for antifungals that are very effective, but also tend to have very severe side effects. They induce autoimmune responses and autoinflammatory responses. We're trying to minimize those, and we're trying to understand the basis of those responses while at the same time maintaining the efficacy of the drug. So the goal here is to probe those inflammatory responses and the autoimmune responses by raising some antibodies to 8 chemokines. So we're probing 8 different molecules on this antibody array, and we array those 23 different antibodies out and assay them through a standard ELISA, enzyme-linked immunosorbent, blah, blah, blah. We immobilize our primary antibody on the membrane. That's raised to the cytokine. And then once we've adhered our cytokines from our cellular medium, from our culture medium, then we challenge with another antibody that's linked to horseradish peroxidase. This, of course, is chemiluminescent. That is, it gives us a handle by which to observe the presence or absence of a chemokine, or a cytokine. So the key here, of course, is that the culture medium is probed for the induction of these autoimmune and inflammatory compounds. And what we see here is that when we challenge the cells with a variety of these antifungals, we can rank them according to, in this case, not efficacy, but how strongly they stimulate the production of these cytokines. And now let's remind ourselves that no longer are we looking at gene expression as a measure of change in the cell, but now we're looking at the level of the presence of the protein itself. And what we see is that for certain of these cytokines, we see a massive increase in the expression over the controls. Now what I'm going to point you to here on the slide is a couple of details. Here in the control, of course, we haven't been challenged with any drug. Let's focus here. This particular duplicate spot happens to correspond to TNF alpha, and this particular duplicate spot happens to correspond to MCP1, which stands for monocyte chemoattractant. I'll write that down. Monocyte chemoattractant 1. And we see that in this case we have massively increased the expression, the presence of that cytokine in the cell after presentation of the antifungal. And in this case we see smaller but clearly measurable changes in that expression level. If we look here at MCP1, and I don't see TNF alpha on this slide, but if we look at MCP1, we can see that here for fungizone, that's a common, the standard formulation of the go-to antifungal. We see we see a massive increase in the presence of that protein compared to these other drugs. And in fact, we can turn to other antifungals, ambisome, which is one that happens to have the fewest side effects, and we see that molecule does not induce the expression of that particular cytokine. So the point being here that we can make correlations with the clinical observations of toxicity, that is to say, fungizone bad, ambisome good, and we try to interpret that in terms of the physical response of the cells. So now we can be systematic in our interpretation of clinical observations. And the question is, how are we going to apply this? So that's another question that I present to you, is these are tools that exist. The question is, what is the clinical utility and or utilization of these? One more time, these kinds of arrays provide another avenue for exploring the adverse drug responses, in this case inflammatory or autoimmune responses of a particular drug treatment regimen. So another tool in the ADR toolbox. But let's turn to something a little bit more complicated now. This is a proteome chip for yeast. In this case, all the S-CRVCI open reading frames are represented, so 6,500 proteins that are spotted as GST fusions on a single slide. Very small amounts of material are required. And on this top slide here, we see the slide probed with an anti-GST antibody labeled with Cy5. So we're looking at that, again, that red fluorescence of Cy5, and in this top slide, really all we're looking at is the relative abundance of any one of those proteins, just giving you a sense of the variability in terms of the application of those proteins. in this, and then, so no data here. This is just control, right? We'll come to the data in just a second. In this inset here, we're looking at a gel. So these guys or gals, I can't remember which jus happens to be, doesn't matter. They took a representative subset and used electrophoresis to probe how well they had actually deposited cells on, deposited proteins on their slides, and they see that things worked okay. And I'm not going to go into the details of how well or poorly they were able to spot, but suffice it to say that on average, there was enough spotting to get reasonably reproducible results. Now, so that's this control. So much for the control. What about challenging this with looking for real protein-protein interactions? And what they did then was label calmodulin with Cy3 and challenge the chip there. And interestingly, they found calmodulin bound to 6 known targets. There are other targets that were known calmodulin ligands that were not bound for various reasons. I don't want to focus on that. Instead, I want to focus on the fact that 33 new targets were observed to bind calmodulin. Why do I mention that? Well, again, we're not learning anything about pharmacogenetics here, but the point is that this is a discovery tool for finding new connections between previously unconnected molecules. So in this case, we have a whole slew of potential targets now that we can think about participating in calmodulin modulated signaling events. So after giving you that yeast example of a protein array, here is the end of the story with the human proteome chip. This is from Invitrogen. This is the proto-array, and now version 5 has some 9,000 proteins spotted on the chip, and these are unique proteins. Here's a sort of a breakdown of where those fall, and you can think about the importance to understanding signaling for example. But also, let's see, these are just by way of introduction, these are just the same idea, GST linked fusion proteins, and this is just a anti-GST Cy5 labeled blah 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 to show you which spots are labeled to what extent. The literature claims that they can detect as much as 25 picograms, which is extremely small amount of protein. We're looking at the costs here per array, so it's not a cheap tool. But what I'd like us to think about in our discussions this week is the value of this kind of a tool as a discovery, particularly in a discovery tool for pharmaco blah 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 omics. So think about that, see what kind of literature you can find to bring to this discussion, and we'll see you on the discussion boards.